Hello everyone, this is chapter 17. It kind of goes along with the DNA chapter, chapter 14. But this is about DNA technology. There are only really 3 slides we need to look at. The rest you can ignore. There are 3 main concepts I want to talk about. So the first concept I want to talk about is gel electrophoresis. You can kind of see the outline of the gel up here. It goes down here. And then goes across here. But that's the gel. These gels are typically about, I don't know, not even a full centimeter deep. But they're usually about this size that I've outlined. Here you can see the outline of the wells, and these wells are little pockets in the gel. These pockets don't go all the way through the gel. They just go partially through the gel. But this is a little reservoir to put your DNA sample in. These gels are typically made out of agarose, which is a derivative of agar. Agar comes from seaweed. It's a thickening agent. It will polymerize whenever you heat it. So in order to make one of these gels, I would just weigh out, let's say I want to make a 1% gel. I'd weigh out 1 gram, add it to 100 milliliters of tris buffer, and I would microwave it. And as soon as the agarose was completely dissolved, I would let the sample cool down and I'd pour it into my mold, and my mold would sort of have these dimensions that I've outlined in red, and then they would have a set of combs that would give you the shape of the well. Once it got cool enough to the touch to pour, I would pour it and into the mold, and that would be it. It would solidify after a minute or two. Inside the gel are a bunch of polymers of agarose. So think of a human traveling through a dense forest and a car traveling through a dense forest. A human traveling through a forest could eventually find a pathway through the trees. But a car would have to sort of drive around until it found like a thinning in the trees or a road. The car would take a lot longer to get through a dense forest, unless there was a road or something like that. And this is kind of the same idea. These gels allow smaller fragments of DNA to travel faster through them, because they can fit through all the tiny holes in this gel matrix. Larger pieces of DNA have to fit through lengthwise, so they travel through the DNA gel slower. So what gel electrophoresis does, the purpose of it is to separate fragments of DNA based on size. So each of these lanes have a DNA sample. It looks like it's been cut by some sort of restriction endonuclease. And then these lanes on the outside have a ladder. This ladder has different bands at known sizes. I'm willing to bet money this super bright one right here is 1,000 kilobases, or 1 kilobase, so it'd be 1,000 base pairs. This bright one here. And these below would be less than that, like maybe 7,500, or not 7,500, 750, and then 500, and then maybe 200 and 100. And so these sizes are based on how many bases there are, so how many nucleotides there are in these fragments. So assuming that, let me pick another color. Assuming that this band is 500 base pairs, that's standard, we know that this fragment in our sample is about 500 base pairs long. So if you run the standards in your samples, you can tell how big your fragments are in your samples by comparing them to the standards. Now the tris buffer part of gel electrophoresis, it makes sure that DNA stays negative and it makes the liquid conductive. When you put this gel into electrophoresis chamber, it's going to use electricity to pull DNA through the gel, and that is because DNA is negatively charged, and it will travel towards the positive electrode if you put electrodes on either side of this gel. So up here where I've drawn the long yellow line up here, you would have the negative electrode. And down here, that blue line is not very dark. Where I've drawn this white line would be the negative electrode. Your DNA will travel away from the white line's the positive electrode. So your DNA would travel away from the yellow electrode at the top, the negative one, and it will travel towards the positive one. That's important. If I had flipped my gel around, that would be bad because your DNA would go this way instead, and it would probably run off your gel before you realized what had happened. So when we do gel electrophoresis in lab, do keep in mind that we're running a current through the liquid. But that's pretty much it. So the purpose of gel electrophoresis is to separate DNA by size. We use electricity to pull DNA through this gel matrix. Smaller DNA fragments will run faster, and large DNA fragments will run slower. So faster fragments will be more towards the bottom. So this fragment is smaller than this fragment. And DNA travels away from the negative electrode and towards the positive electrode. So that's pretty much it for gel electrophoresis. Let's move on to the next topic. So next topic is polymerase chain reaction, PCR. The purpose of PCR is to copy DNA, so make lots and lots of copies of DNA, which is also known as amplifying. So when you amplify sound, you take a quiet sound and you make it louder. When you amplify DNA, you take a little bit of DNA and you make a lot. The basis for PCR is that a PCR program will run through many cycles. Each cycle has the same 3 steps, and we'll talk about those 3 steps. And each cycle you're going to double your DNA content. So if you have 1 copy of the gene before cycle 1, you would have 2 copies of the gene after cycle 1. If you have 2 copies of the gene before cycle 2, you had 4 copies after cycle 2. If you have 4 copies before cycle 3, you would have 8 copies. And so this DNA is going to increase exponentially. We've gone from 2 to 8 copies in 3 cycles. So let's talk about what each cycle entails, and then we'll talk about what goes into our PCR tube. So the first step in the PCR cycle is denaturation. Denaturation is a way to get DNA from being double-stranded to single-stranded. So in DNA replication, helicase first had to separate the two strands so that they could act as templates. The exact same thing has to happen in polymerase chain reaction. The difference is instead of using helicase, we're just using heat. So if we heat this sample up to 95 to 99 degrees Celsius, we will break the hydrogen bonds, but the covalent bonds will remain intact. So our strands will remain intact. My mouse is not working very well today. But they will be single-stranded instead of double-stranded. We will have broken those hydrogen bonds. So the next step is annealing. Annealing. Annealing means to join. So in PCR, we typically don't copy the entire template. Let's say we just took some cheek cells from me and took my entire genome, all 46 chromosomes. In PCR, you typically would not copy every single bit of all 46 chromosomes. You typically amplify just something you want. And the shorter segment of DNA, the better, because then your cycle times can be shorter. So let's say for this PCR, we're just amplifying this red gene. In order to amplify this red gene, you would design primers, and primers are little, you've heard the word primer in DNA replication, and we need primers in order for our DNA polymerase to work. The same thing is true in PCR. Now, in DNA replication, primers are made from RNA. In PCR, they're made from DNA. So these green things right here, there and there, are primers, and they are made out of DNA. The annealing step, you cool down your test tube from a 95 to 99, which we did for annealing, down to somewhere in the 50 to 65 degrees Celsius range. I've seen lower than that and maybe a little bit hotter than that, but those programs typically, those PCR programs typically do not work quite as well. So typically you want your primers to anneal between 50 and 65 degrees Celsius, so you just cool your test tube down to that temperature and your primers will bind to the single-stranded DNA. Notice this DNA is still single-stranded. So the last step, I've seen it called elongation, I've seen it called synthesis, but basically polymerase, DNA polymerase, will bind to the primer and elongate off of the end of that primer using the single-stranded DNA as a template. And the same thing will happen on the other strand. So polymerase will bind to the primer and elongate off of that primer based on the 5' to 3' direction. Okay, so those are the 3 steps. Oh, and for the DNA synthesis to occur, I believe for Taq polymerase, it's 72 degrees Celsius. And there are different polymerases that we use, but Taq was the first. And it's still well used. There's synthetic versions now that people swear by, but Taq polymerase is still definitely used. So let's talk about Taq polymerase for a minute, because Taq polymerase is the reason that we were able to develop PCR. So the problem with human polymerase is that it works at roughly 42 degrees Celsius. And that is below a temperature where primer annealing could occur. So primer annealing, remember I said, happens between 50 and 65 degrees Celsius. If we cooled this test tube down to 42, where human DNA polymerase works, our DNA would actually go back to being single-stranded. You would not get elongation. You would not get DNA synthesis. So, no one could ever develop polymerase until someone started studying this bacterium called Thermus aquaticus. And this bacterium were in hot geysers at Yellowstone National Park. And because these bacteria could survive at very hot temperatures, their DNA polymerase could work at these high temperatures. So instead of working at 42 degrees Celsius, this Taq polymerase worked at 72 degrees Celsius, which was higher than your typical annealing temperature for primers. So now we're in business. Now we can do PCR, and the way you do it is just by controlling the temperature. So your DNA synthesis step can now be done at 72 degrees instead of 42 degrees, and polymerase chain reaction works. So these cycles start off at 95 to 99 degrees for denaturation, that's making DNA single-stranded. It drops back down to 50 to 65 degrees Celsius. That's so that annealing can happen. Your primers bind to the single-stranded DNA template. And then we'll heat this solution back up to 72 degrees Celsius to get the elongation from the Taq polymerase. All right, so those are your cycles, and every time you double whatever you're amplifying. So every cycle you double what you're amplifying. So let's talk a little bit about what goes into a PCR tube. So we need the template, and this template can be genomic DNA, it can be synthetic. By genomic, that would probably be isolated from some live tissue, such as cheek cells or maybe hair follicles. Template. But your template will provide the template for DNA synthesis. It will have your gene of interest or whatever you're trying to amplify. The other thing you need is a set of primers. And the primers give you the specificity. Your primers determine what part of all this DNA template are you going to amplify. And that could be 2,000 base pairs, it could be 5,000 base pairs, it could be smaller, it could be 200 base pairs. But your primer determines what part of the DNA template you are amplifying, that you're copying. You need deoxynucleotide triphosphates, DNTPs. And these are your nucleotides that are the building blocks for DNA so that you can make DNA. And then you need some sort of buffer. This buffer keeps your enzyme happy, keeps your DNA happy. It just provides the right environment for everything to work. Mainly pH, salt concentration, magnesium concentration, things like that. And then lastly, you need your polymerase. In lab, we're going to use Tech. I can do a Q, I can do a Q. All right. Is there anything else we need? We need template, primer, DNTPs, buffer, and Tech. Nope, that's pretty much it. So then you would close your tube, pop. You would put all these in your tube and then close the lid on the tube and then put it in this machine called a thermocycler, and it's just going to change the temperature. So we're going to do 95 to 99 for denaturation, 50 to 65 for annealing, and then 72 degrees for DNA synthesis. Now, before you actually get, before and after your cycles, oh, and typically you can run, I don't know, I've seen as little as 15 cycles. I've seen as many as like 40 cycles. So it kind of depends on how much DNA you start with and how much you need to end up with. When we do our PCR program in lab, we do 35 cycles, so that's quite a few cycles. Before you do a cycle, before you do all your cycles, you do an extra annealing step, and you do that for like 10 minutes just to make sure all your template is single-stranded. And then after the end of all your cycles, you do an extra elongation step, or what this book calls DNA synthesis step. So you do 72 degrees for an extra 10 minutes, just to make sure that all your pieces of DNA that you're trying to synthesize are fully elongated. So that's PCR. Its main purpose is to copy DNA. Primers provide the specificity. TAC is special because it works at a high temperature, and we needed that high temperature for PCR to work. PCR is it's a cycle with 3 steps. The first step is denaturation. It's done at 95 to 99 degrees Celsius. You're making DNA single-stranded. The second step of the cycle is annealing. That's between 50 and 65 degrees Celsius. You're allowing your primers to bind to the template. And then the third step, elongation, or the synthesis step, DNA polymerase, Taq polymerase will elongate off the primer using the template. And then also in your test tube, you need DNTPs, the building blocks of DNA, so you can synthesize this DNA. And then you need buffer to keep Taq polymerase happy and your DNA happy. So that is it for PCR. The last thing I want to talk about is digesting and ligating DNA. Let's say you want to engineer some bacteria that can make human insulin. Human insulin is an expensive drug. It would be great if we could get bacteria to grow it for us. Bacteria multiply. Some bacteria can have a generation time of 20 minutes. So they can double their numbers in 20 minutes, which is amazing. Humans cannot do that. And bacteria are easy to feed. They just need some glucose and some broth, some amino acids, and they're happy. So bacteria are cheap and easy, and it's easy to get lots of them. So it would be wonderful to have some bacteria that could grow human insulin. So how would we get a bacteria to make human insulin. Well, bacteria, we've talked about them quite a bit. They have a single circular chromosome, but I've also mentioned at least once that sometimes they have these little circular pieces of DNA called plasmids. And bacteria will carry around plasmids that are beneficial to them. Like a lot of these bacteria superbugs that you've heard about that have antibiotic resistance, they got that antibiotic resistance from a plasmid. This plasmid, like I said, is extra chromosomal, so it's separate from the bacterial chromosome, but it replicates whenever the chromosome does. So they're sort of self-replicating little circular pieces of DNA. And we've identified and actually manufactured, by we I mean scientists, certain plasmids that we can use, and they have these areas called cloning sites, where there's lots of places for us to cut it open with different restriction endonucleases. So what you do is you get some human DNA and you use PCR to amplify up these little what we call cassettes. So this would be the human insulin gene, and then on either side of it you would have a site for a restriction endonuclease cut. You would digest your cassette and your plasmid with the same restriction endonuclease. That means their sticky ends would be complementary to each other. So you take some cassette DNA that's been digested and put it into a test tube. You take some plasmid DNA that's been digested, put that in the same tube, and then you add an enzyme that you've heard of before called ligase. Oops. It's a funky E, but that's an E. And ligase will glue the two pieces together. So you have now glued together your plasmid, which you need the plasmid because it will self-replicate inside the bacterium. And we've glued this little cassette into our plasmid that has the human insulin gene in it. So bacteria, if they get stressed out, will take up foreign DNA from the environment, just on the off chance that it is helpful to them in order to survive the environment. And the way you do this is you heat shock them. So you put them on ice. You would mix your bacteria on ice with your plasmid that you've created with human insulin gene inside it. And then you'd transfer them straight to a hot bath, a water bath. This heat shocks them, they freak out a little, and then they take up foreign DNA. And this way we would get the plasmid into our bacteria. So the plasmid would go from outside the bacteria into the bacteria. Now, I don't know if you can read this, but I believe it says ampicillin, or AMP for ampicillin, and is the ampicillin resistance gene. So after you heat shot your bacteria, you grow them up a bit in a broth, and then you plate them on an agar plate with ampicillin. And ampicillin is an antibiotic. It will kill most bacteria. And but if these bacteria have our plasmid with the human insulin gene in it, this also gives them ampicillin resistance. So if you plate them on a plate with ampicillin, this is known as selection. The bacteria that do not have the plasmid will die due to the ampicillin. The bacteria that do have the plasmid with the human insulin gene will survive on the plate of ampicillin. So this way we've sort of forced our bacteria to hang on to our plasmid with the human insulin gene on it. Pretty smart, huh? So now you just take one of these colonies from the ampicillin plate, you grow it up. When they grow, they make human insulin, and then you lyse all the cells and harvest the human insulin from them, purify that out, and then you got human insulin you can inject in people with type 1 diabetes. So this is why molecular cloning is so important to us. It's a very powerful tool. And it's very controllable. And once you learn how to do it, it's quite fun. I did it a couple times. Anyway, those are the 3 major topics I want you to know about. Gel electrophoresis, PCR, and molecular cloning. Cloning is really how we use restriction endonucleases quite a bit more than in forensics. In forensics now, we typically do a DNA fingerprint, which uses PCR. It doesn't use the fragmentation of DNA. So that's it for this chapter. The next chapter you should do is chapter 15, which is transcription translation. Anyway, good luck.